Hello there, and welcome to the Sleepy Science Society. I'm so glad you found your way here tonight. Perhaps you had a long day, or maybe you're the kind of a person who finds peace in understanding the hidden machinery of existence. Either way, you're in the right place. Before we begin our journey into one of life's most fundamental miracles, if these explorations help you unwind or spark your curiosity as you drift toward sleep, consider liking and subscribing to the channel. It helps others discover these quiet contemplations too, one restful mind at a time. Tonight, we're venturing into something extraordinary, something happening inside you right now, at this very moment, in trillions of places simultaneously. We're exploring cellular respiration, the elegant molecular dance that transforms the food you ate today into the energy that powers every thought, every heartbeat, every breath. It's a process so fundamental that it connects you to every living thing on this planet, from the smallest bacterium to the largest whale. And yet, it happens in near silence, in spaces too small to see, following choreography that has been perfected over billions of years. You don't need to do anything but rest. Let your breathing slow and your mind settle. Because what we're about to explore is already happening inside you, a quiet miracle unfolding in the darkness of your cells. It's the process that allows life to continue, moment by moment, breath by breath. And though the details may be complex, the story itself is one of transformation, of ancient pathways, and of the incredible ingenuity written into the very fabric of living things. The energy currency of life. Deep within each of your cells, in chambers smaller than you can imagine, a remarkable transaction is taking place. Energy is being converted, transferred, stored, and released in a cycle that never stops from the moment of your first breath until your last. This is the business of cellular respiration, and its currency is a molecule with a rather plain name: adenosine triphosphate, or simply ATP. Think of ATP as the universal energy coin of living cells. Just as you might use money to purchase different goods and services, your cells use ATP to pay for virtually everything they do. Every muscle contraction that lets you turn a page or walk across a room. Every electrical impulse that carries a thought through your brain. Every chemical reaction that builds new proteins or repairs damaged DNA requires payment in ATP. Your heart beats about 100,000 times each day, and every single contraction is powered by ATP. Your brain, accounting for only about 2% of your body weight, consumes roughly 20% of your body's total energy budget, burning through ATP molecules at an astonishing rate as neurons fire and communicate. The truly miraculous part is how much ATP your body produces. On an average day, a typical human will create and consume roughly their own body weight in ATP. A person weighing 70 kilograms will cycle through approximately 70 kilograms of ATP in a single day. But here's the remarkable thing: you don't carry 70 kilograms of ATP around with you. Instead, you are constantly recycling the same molecules, breaking them down, and rebuilding them hundreds of times each day. Each ATP molecule might be recycled every few minutes, a continuous assembly and disassembly that never pauses. The structure of ATP itself is elegantly simple. Imagine a small molecule consisting of adenosine, a structure made from a nitrogen-containing base attached to a 5-carbon sugar, with a tail of 3 phosphate groups strung together like pearls on a string. These phosphate groups are the key to ATP's power. The bonds connecting them, particularly the bond between the second and third phosphate groups, store considerable energy. When your cell needs energy for any task, it breaks that bond, releasing the third phosphate group and converting ATP into ADP, adenosine diphosphate, a molecule with only 2 phosphate groups instead of 3. This releases energy that the cell can immediately use to drive chemical reactions, transport molecules across membranes, or generate the mechanical forces needed for movement. But this raises an obvious question. If your cells are constantly breaking down ATP into ADP and phosphate to release energy, how do they avoid running out? The answer is cellular respiration, the process that takes ADP and phosphate and reassembles them back into ATP, recharging the molecular battery. And the energy for this recharging doesn't appear from nowhere. It comes from the food you eat, the molecules of carbohydrate, fat, and protein that you consume each day. Cellular respiration is the bridge between the chemical energy locked in the bonds of food molecules and the universal energy currency your cells can actually use. The process we're about to explore in detail is primarily focused on one particular food molecule: glucose, a simple sugar that serves as the primary fuel for most cells. Glucose is a 6-carbon molecule with the chemical formula C6H12O6, and within its structure lies stored energy originally captured from sunlight by plants through photosynthesis. When you eat an apple or a slice of bread, the carbohydrates are eventually broken down into glucose molecules that enter your bloodstream. These glucose molecules are then delivered to your cells, where cellular respiration extracts their stored energy and uses it to regenerate ATP from ADP and phosphate. The complete oxidation of one molecule of glucose releases a substantial amount of energy, enough to drive the synthesis of up to 36 or 38 molecules of ATP, depending on the exact conditions and efficiency of the process. This might not sound like much until you consider the scale. Every second, trillions of glucose molecules are being processed through cellular respiration in your body. The energy flows continuously, like a river that never runs dry, because you keep eating and breathing, supplying the raw materials, glucose and oxygen, that the process requires. Cellular respiration is often summarized by a deceptively simple chemical equation. 1 molecule of glucose plus 6 molecules of oxygen yields 6 molecules of carbon dioxide, 6 molecules of water, and energy in the form of ATP. Written out, it looks like this: 6H12O plus 6O2 yields 6 CO2 plus 6 H2O plus energy. But this equation, while accurate, tells you almost nothing about the astonishing complexity of what's actually happening. It's like reducing a symphony to a single note, or describing an entire journey by naming only the starting point and destination. The real story of cellular respiration involves multiple stages, dozens of enzymes, countless intermediate molecules, and a series of precisely choreographed events that occur in different compartments of the cell. To truly appreciate this process, we need to understand where it happens, and that brings us to one of the most fascinating structures in the biological world, the mitochondrion, the cellular powerhouse, mitochondria. If you could shrink yourself down to microscopic size and wander through the interior of one of your cells, you would find yourself in a crowded, bustling environment. The cell is not an empty bag of fluid, but a highly organized city of structures, each performing specific tasks. Among these structures, none is more critical to energy production than the mitochondria, often called the powerhouses of the cell, and the nickname is well deserved. Mitochondria are small, bean-shaped or elongated structures, typically 1 to 10 micrometers in length, scattered throughout the cell's interior. Most human cells contain hundreds or even thousands of mitochondria, with the exact number depending on the cell's energy demands. Cells that work hard, like muscle cells and heart cells, are packed with mitochondria because they need enormous amounts of ATP to fuel their constant activity. A single heart muscle cell might contain 5,000 mitochondria, occupying up to 40% of the cell's total volume. Even less active cells have plenty of mitochondria, quietly generating the ATP needed for basic cellular housekeeping. What makes mitochondria particularly fascinating is their structure. Unlike many other cellular compartments, mitochondria are enclosed by not one but two membranes, an outer membrane and an inner membrane, each with distinct properties. The outer membrane is relatively smooth and porous, allowing many small molecules to pass through. The inner membrane is quite different. It's highly folded into structures called cristae, which look like the pleated folds of an accordion. These folds dramatically increase the inner membrane's surface area, providing more space for the molecular machinery of cellular respiration to operate. If you could unfold all the inner membranes from all the mitochondria in a single liver cell, they would cover an area of several square meters. The two membranes create two distinct compartments within the mitochondrion. The space between the outer and inner membranes is called the intermembrane space, while the interior enclosed by the inner membrane is called the matrix. These compartments are not arbitrary divisions. They are essential to how cellular respiration works. Different stages of the process occur in different locations, and the inner membrane itself serves as a critical platform for the final, most productive stage of ATP synthesis. Inside the matrix, you would find a complex mixture of enzymes, dissolved molecules, and even some DNA. Yes, mitochondria have their own genetic material, separate from the DNA in the cell's nucleus. This fact is a clue to one of biology's great mysteries: the origin of mitochondria. The prevailing theory, supported by extensive genetic evidence, is that mitochondria were once free-living bacteria that entered into a symbiotic relationship with the ancestors of modern eukaryotic cells, perhaps 2 billion years ago. These bacteria were capable of aerobic respiration, the process of using oxygen to extract energy from food molecules. When they took up residence inside other cells, they provided their hosts with a tremendous energetic advantage. allowing those cells to produce far more ATP than was possible through older, less efficient metabolic pathways. Over countless generations, the partnership became permanent. The bacteria evolved into mitochondria, losing many of their original genes but retaining their essential function, generating ATP through cellular respiration. This ancient history is still visible today. Mitochondrial DNA is circular, like bacterial DNA, and quite unlike the linear chromosomes found in the cell nucleus. Mitochondria replicate independently of the cell's division cycle, splitting in two like bacteria. And the inner mitochondrial membrane contains unique lipids more typical of bacterial membranes than of eukaryotic cell membranes. When you look at a mitochondrion, you're seeing an evolutionary partnership that has endured for eons, a collaboration so successful that virtually all complex life on Earth depends on it. The mitochondrion's structure is beautifully suited to its function. The matrix houses the enzymes for one of cellular respiration's key stages, the Krebs cycle, which we'll explore in detail shortly. The inner membrane, with its vast folded surface area, holds the protein complexes that make up the electron transport chain and ATP synthase, the molecular machines responsible for producing the majority of the cell's ATP. The intermembrane space serves as a reservoir for hydrogen ions, creating an electrochemical gradient that powers ATP synthesis in a process we'll come to understand as chemiosmosis. Everything about the mitochondrion's architecture reflects its purpose: to extract as much energy as possible from glucose and other fuel molecules and to convert that energy into ATP. And the process begins not in the mitochondrion at all, but in the cell's main fluid-filled interior, the cytoplasm, with a stage called glycolysis. Stage 1. Glycolysis, the ancient pathway. Long before oxygen was abundant in Earth's atmosphere, before mitochondria existed, life found a way to extract energy from sugar molecules. That ancient metabolic pathway still operates today in virtually every living cell on the planet. It's called glycolysis, meaning the breaking of glucose, and it's the first stage of cellular respiration. Glycolysis occurs in the cytoplasm, the gel-like fluid that fills the cell outside of the membrane-bound organelles. It doesn't require oxygen, which is why it's sometimes called the anaerobic stage of cellular respiration. This anaerobic nature is a relic of glycolysis's ancient origin, dating back to a time when Earth's atmosphere contained little or no oxygen. The fact that this pathway still functions in oxygen-rich environments, and that it serves as the gateway to more efficient aerobic processes, speaks to evolution's tendency to build upon existing structures rather than replacing them entirely. The goal of glycolysis is straightforward. Take one molecule of glucose, a 6-carbon sugar, and break it down into 2 molecules of pyruvate, a 3-carbon compound. Along the way, the process captures some of the energy released and uses it to produce a small amount of ATP and to fill electron carrier molecules with high-energy electrons. The entire sequence involves 10 distinct chemical reactions, each catalyzed by a specific enzyme, each transforming the molecule in a precise way. It's a carefully orchestrated metabolic pathway, and while the details can seem intricate, the overall arc of the story is clear. Glycolysis can be divided into two phases: the energy investment phase and the energy payoff phase. The names tell you exactly what's happening in each half of the process. In the energy investment phase, which encompasses the first 5 steps of glycolysis, the cell actually consumes ATP rather than producing it. This might seem counterintuitive. Why would a process designed to generate energy start by spending energy? But it makes sense when you consider what's being accomplished. Glucose is a stable molecule. To break it apart and access its stored energy, you need to destabilize it first, to prime it for the reactions to come. This priming requires an input of energy. The process begins when an enzyme transfers a phosphate group from ATP to glucose, converting it into glucose 6-phosphate and leaving behind ADP. This addition of a phosphate group has 2 effects. First, it traps the glucose inside the cell because the phosphorylated form cannot easily cross the cell membrane. Second, it makes the molecule more reactive, less stable, and more amenable to further chemical transformations. The glucose 6-phosphate is then rearranged into a slightly different 6-carbon sugar called fructose 6-phosphate, which is then phosphorylated again by another ATP molecule, producing fructose 1,6-bis phosphate, a 6-carbon sugar with phosphate groups attached at both ends. This doubly phosphorylated molecule is now unstable enough to be split. An enzyme cleaves it into 2 3-carbon molecules, each carrying a phosphate group. These molecules are quickly interconverted, and both follow the same pathway from this point forward. Because the original 6-carbon glucose has been split into 2 3-carbon units, everything that happens from here on effectively happens twice for each glucose molecule that enters glycolysis. This doubling is important to keep in mind as we tally up the energy yield. The energy investment phase has consumed 2 molecules of ATP per glucose, but now the payoff begins. In the energy payoff phase, the 2 3-carbon molecules, now called glyceraldehyde 3-phosphate, proceed through a series of reactions that harvest energy. Each 3-carbon molecule is oxidized, meaning electrons are removed from it. These electrons are captured by a carrier molecule called NAD+, which becomes NADH when it accepts a pair of electrons and a hydrogen ion. The oxidation releases enough energy to attach another phosphate group to each 3-carbon molecule, creating intermediates that now carry 2 phosphate groups. These high-energy intermediates can now transfer their phosphate groups to ADP, generating ATP. This direct transfer of a phosphate group from a substrate molecule to ADP is called substrate-level phosphorylation, distinguishing it from the more complex ATP synthesis that will occur later in the mitochondria. Each 3-carbon molecule donates 2 phosphates to ADP over the remaining steps of glycolysis, producing 2 ATP molecules per 3-carbon molecule, or 4 ATP total for the original glucose. By the end of glycolysis, the glucose molecule has been converted into 2 molecules of pyruvate, a 3-carbon compound. The total energy harvest from this process is 2 molecules of NADH, which carry high-energy electrons and a net gain of 2 ATP. Remember, 4 ATP were produced, but 2 were consumed in the investment phase, leaving a net yield of 2 ATP per glucose. It's not a large amount of energy, only about 2% of the total energy available in the glucose molecule. But it's enough to be useful, and crucially, it sets the stage for the much more productive processes that follow. The pyruvate molecules produced by glycolysis stand at a metabolic crossroads. If oxygen is available, they will proceed into the mitochondria and enter the aerobic stages of cellular respiration, where much more energy will be extracted. If oxygen is scarce or absent, they will be diverted into fermentation pathways, which regenerate NAD+, so that glycolysis can continue even without oxygen. We'll explore fermentation later. For now, let's follow the pyruvate molecules into the mitochondria and see what happens when oxygen is present. The transition. Pyruvate oxidation. Before the pyruvate molecules can enter the Krebs cycle, they must be modified. This modification occurs in a brief but important transition reaction, sometimes called the oxidative decarboxylation of pyruvate, or simply pyruvate oxidation. It's a short step, but it's essential for linking glycolysis to the more elaborate machinery of the Krebs cycle. Each pyruvate molecule, fresh from glycolysis in the cytoplasm, is transported across the mitochondrial membranes into the matrix. Once inside the matrix, the pyruvate encounters a large enzyme complex called pyruvate dehydrogenase, a molecular machine consisting of multiple copies of 3 different enzymes, along with 5 different coenzymes that assist in the reaction. This enzyme complex is enormous by molecular standards, one of the largest multi-enzyme complexes in the cell, and its size reflects the complexity and importance of the reaction it catalyzes. The pyruvate dehydrogenase complex performs several transformations simultaneously. First, it removes a carbon atom from pyruvate in the form of carbon dioxide. Pyruvate is a 3-carbon molecule. After decarboxylation, what remains is a 2-carbon fragment. This carbon dioxide molecule is a waste product. In your body, it will eventually be transported to your lungs and exhaled. Every breath you take releases carbon dioxide that was once part of a glucose molecule. A reminder that respiration is not just about generating energy, but also about processing the breakdown products of fuel molecules. The remaining 2-carbon fragment is then attached to a molecule called coenzyme A, often abbreviated as CoA. Coenzyme A is a complex molecule derived from vitamin B5, and it serves as a carrier, a molecular handle that allows the 2-carbon fragment, now called an acetyl group, to be transferred into the Krebs cycle. The resulting molecule, acetyl-CoOA, is one of the central players in cellular metabolism. It's not only a product of glucose breakdown, but also a key intermediate in the metabolism of fats and some amino acids. Acetyl-CoA is, in a sense, the common currency into which many different fuel molecules are converted before their energy can be fully extracted. During the conversion of pyruvate to acetyl-CoA, electrons are also removed from the carbon atoms, and these electrons are captured by another molecule of NAD+, reducing it to NADH. This NADH molecule joins the two produced during glycolysis as carriers of high-energy electrons, electrons that will eventually power ATP synthesis in the electron transport chain. Because each glucose molecule produces two pyruvate molecules during glycolysis. Pyruvate oxidation happens twice per glucose. The total yield from this transition stage is 2 molecules of acetyl-CoA, 2 molecules of NADH, and 2 molecules of carbon dioxide. These acetyl-CoA molecules are now ready to enter the Krebs cycle, a circular metabolic pathway that will extract nearly every last bit of usable energy from the remnants of the original glucose molecule. The transition from glycolysis to the Krebs cycle represents a geographic and biochemical shift. Glycolysis happened in the open waters of the cytoplasm, an ancient pathway that evolved before cells even had specialized compartments. But now we're inside the mitochondria, in the matrix, where the more sophisticated machinery of aerobic respiration operates. The pathways here are more efficient, more complex, and entirely dependent on the presence of oxygen, even if oxygen doesn't directly participate in the Krebs cycle itself. Its role comes later, at the very end of the electron transport chain, but it's essential to keep the whole system running. Without oxygen, the Krebs cycle and electron transport chain grind to a halt. With acetyl-CoA in hand, we're ready to enter one of biochemistry's most elegant constructions, the citric acid cycle. Stage 2. The Krebs cycle. The Krebs cycle, also known as the citric acid cycle or the tricarboxylic acid cycle, is named after Hans Krebs, the biochemist who elucidated much of the pathway in the late 1930s. For this work, Krebs received the Nobel Prize in Physiology or Medicine in 1953. The cycle is a masterpiece of metabolic engineering, a series of 8 chemical reactions that occur in sequence, each feeding into the next, forming a closed loop that regenerates its starting compounds and can therefore turn continuously as long as acetyl-CoA is available. The cycle takes place entirely within the mitochondrial matrix, where all the necessary enzymes float in solution, or are loosely associated with the inner mitochondrial membrane. Each turn of the cycle processes one molecule of acetyl-CoA, extracting its energy and releasing its carbon atoms as carbon dioxide. Since each glucose molecule produces 2 acetyl-CoA molecules, the cycle must turn twice to fully process the carbon skeleton of one glucose. The cycle begins when the 2-carbon acetyl group from acetyl-CoA combines with a 4-carbon molecule called oxaloacetate, forming a 6-carbon molecule called citrate, or citric acid, which gives the cycle one of its names. The formation of citrate is catalyzed by an enzyme called citrate synthase, and it's an irreversible reaction, a committed step that launches the acetyl group on its journey through the cycle. Once citrate is formed, the coenzyme A is released and can return to accept another acetyl group from pyruvate oxidation. Citrate then undergoes a series of transformations, each catalyzed by a specific enzyme. The 6-carbon citrate is rearranged into a slightly different 6-carbon molecule called isocitrate. This rearrangement might seem like a minor reshuffling, but it's essential because isocitrate is susceptible to a type of reaction that citrate is not. In the next step, isocitrate undergoes oxidative decarboxylation. This means two things happen. Carbon dioxide is removed, reducing the 6-carbon isocitrate to a 5-carbon molecule, and electrons are extracted and transferred to NAD+, forming NADH. This step is catalyzed by the enzyme isocitrate dehydrogenase, and it produces alpha-ketoglutarate, the 5-carbon molecule that remains after carbon dioxide leaves. This is the first of several oxidation reactions in the Krebs cycle, and each oxidation captures electrons in the form of NADH or another electron carrier, FADH2, which we'll encounter shortly. The 5-carbon alpha-ketoglutarate is then oxidized and decarboxylated again by a large enzyme complex called alpha-ketoglutarate dehydrogenase, which is structurally similar to the pyruvate dehydrogenase complex we encountered earlier. Another molecule of carbon dioxide is released. Another pair of electrons is captured by NAD+ to form NADH, and the remaining 4-carbon fragment is attached to coenzyme A, forming succinyl-CoA. At this point, all 3 carbon atoms from the original acetyl group have been released as carbon dioxide. The carbon atoms you exhale are the remnants of the glucose molecules you consumed, fully oxidized and leaving the cell as metabolic waste. The cycle now focuses on regenerating oxaloacetate so that the cycle can continue. The 4-carbon succinyl-CoA is converted to succinate, and in the process, the energy stored in the bond between succinate and coenzyme A is captured to form a molecule called GTP, guanosine triphosphate, which is energetically equivalent to ATP. In some organisms or cell types, ATP is produced directly in this step. In others, GTP is produced and can easily be converted to ATP. Either way, this is another example of substrate-level phosphorylation, a direct transfer of a phosphate group to form a high-energy molecule. Succinate, a 4-carbon molecule, is then oxidized to fumarate, another 4-carbon molecule, but this time the electrons are not accepted by NAD+. Instead, they are captured by a different carrier called FAD, flavin adenine dinucleotide, which becomes FADH2 when it accepts a pair of electrons and 2 hydrogen atoms. FADH2 is similar to NADH, but carries its electrons at a slightly lower energy level, a difference that will matter when these carriers deliver their electrons to the electron transport chain. Fumarate is then hydrated, meaning a molecule of water is added to it, forming malate. And, finally, malate is oxidized to oxaloacetate, with the electrons captured by yet another molecule of NAD+, forming NADH. The cycle has come full circle. Oxaloacetate is back, ready to accept another acetyl group from acetyl-CoA, and the cycle can turn again. Let's pause and take inventory of what one turn of the Krebs cycle produces. Starting with 1 acetyl-CoA, The cycle releases two molecules of carbon dioxide, the carbon atoms from the acetyl group fully oxidized and ready to leave the cell. It generates three molecules of NAEA, carrying 6 high-energy electrons. It produces one molecule of FADH2, carrying 2 high-energy electrons. And it generates one molecule of ATP, or its equivalent GTP, through substrate-level phosphorylation. Remember, the cycle turns twice for each glucose molecule, so the total yield per glucose is 4 carbon dioxide, 6 NADH, 2 FADH2, and 2 ATP. The carbon dioxide diffuses out of the mitochondria and eventually out of the cell. In your body, it's carried by the bloodstream to the lungs, where you exhale it. The ATP produced in the Krebs cycle is useful, but it represents only a small fraction of the glucose molecule's total energy. The real treasure lies in the NADH and FADH2 molecules. These electron carriers are loaded with high-energy electrons extracted from the chemical bonds of glucose, and those electrons are about to enter the most productive stage of cellular respiration, the electron transport chain, where the bulk of the cell's ATP will be generated. The Krebs cycle is often depicted as a circular diagram, a wheel that turns continuously, feeding on acetyl-CoA and producing carbon dioxide, electron carriers, and a modest amount of ATP. But it's more than just a wheel. It's a hub, a central metabolic intersection. The intermediates of the Krebs cycle are starting points for many biosynthetic pathways. Alpha-ketoglutarate can be converted into amino acids. Succinyl-CoA is a precursor for heme, the molecule that carries oxygen in your red blood cells. Oxaloacetate can be used to synthesize glucose in a process called gluconeogenesis. The Krebs cycle is not merely a pathway for breaking down molecules, but a central node in the cell's metabolic network, linking energy production to biosynthesis and providing the building blocks for countless other processes. And now, with the Krebs cycle complete and the carbon skeleton of glucose fully dismantled, we turn to the electron transport chain, where the energy carried by NADH and FADH2 will be harnessed to generate the vast majority of the cell's ATP. Stage 3. The electron transport chain. If glycolysis and the Krebs cycle are the opening movements of a symphony, the electron transport chain is the grand finale, a crescendo of molecular activity that produces the majority of the ATP generated by cellular respiration. It's here, embedded in the inner mitochondrial membrane, that the high-energy electrons carried by NADH and FADH2 are finally put to work, driving the creation of a proton gradient that will power ATP synthesis. The electron transport chain is not a single pathway, but a series of protein complexes, each containing clusters of metal ions and other cofactors that can accept and donate electrons. These complexes are embedded in the inner mitochondrial membrane, surrounded by a lipid bilayer that is impermeable to ions, a critical property for the process to work. There are 4 main complexes, imaginatively named Complex E, Complex 2, complex 3 and complex 4, along with two mobile electron carriers, coenzyme Q and cytochrome C, that shuttle electrons between the complexes. The entire system works like a molecular assembly line, passing electrons from one carrier to the next, each step releasing a small amount of energy. The journey begins when NADH or FADH2 delivers its electrons to the electron transport chain. NADH, which carries electrons at a relatively high energy level, donates its electrons to complex Y, also known as NADH dehydrogenase. This is a massive protein complex, one of the largest in the entire respiratory chain, consisting of more than 40 different protein subunits arranged in an L-shaped structure. Within complex Y, the electrons from NADH are transferred through a series of iron-sulfur clusters, small groups of iron and sulfur atoms that can accept and release electrons. As the electrons move through complex 1, the energy they release is used to pump protons from the mitochondrial matrix across the inner membrane into the intermembrane space. For every pair of electrons that passes through complex Y, approximately 4 protons are pumped across the membrane. Meanwhile, FADH2, which carries its electrons at a slightly lower energy level than NADH, delivers them to complex 2, also called succinate dehydrogenase. You might recognize this enzyme. It's the same enzyme that catalyzes the conversion of succinate to fumarate in the Krebs cycle. A nice example of how biochemical pathways are interconnected. Complex 2 does not pump protons across the membrane. Instead, it simply passes the electrons from FADH2 to coenzyme Q, a small, lipid-soluble molecule that floats within the inner mitochondrial membrane. Coenzyme Q, also known as ubiquinone, is a mobile electron carrier that collects electrons from both complex 1 and complex 2 and transports them to complex 3. Complex 3, or cytochrome BC1 complex, accepts electrons from coenzyme Q and passes them on to another mobile carrier, cytochrome C, a small protein that floats in the intermembrane space and varies electrons from complex 3 to complex 4. As electrons flow through complex 3, more protons are pumped across the membrane, approximately 4 protons for every pair of electrons. The exact mechanism of proton pumping in complex 3 is intricate, involving a process called the Q cycle that allows complex 3 to pump more protons than it could otherwise. Cytochrome C delivers the electrons to complex 4, also known as cytochrome C oxidase, the final complex in the electron transport chain. Complex OVHU is a remarkable molecular machine that catalyzes one of the most important reactions in all of biology, the reduction of oxygen to water. This is where oxygen, the oxygen you breathe, finally enters the story. Molecular oxygen, OCU, is the final electron acceptor in the electron transport chain. Each oxygen molecule can accept 4 electrons, and when it does, it combines with 4 protons to form 2 molecules of water. This reaction is exquisitely controlled. Oxygen is highly reactive, and if its reduction were not carefully managed, it could produce dangerous reactive oxygen species that damage cellular components. Complex IV ensures that the reduction proceeds smoothly and safely. As electrons pass through complex ivo to oxygen, additional protons are pumped across the membrane, contributing further to the proton gradient. The cumulative effect of all this electron transport and proton pumping is the creation of a substantial proton gradient across the inner mitochondrial membrane. The intermembrane space becomes acidic, with a high concentration of protons, while the mitochondrial matrix remains relatively basic, with a low concentration of protons. This gradient has two components: a concentration gradient, more protons on one side of the membrane than the other, and an electrical gradient, a charge difference because protons are positively charged. Together, these gradients form an electrochemical gradient, also called the proton motive force, and it represents a form of stored potential energy, much like water held behind a dam. The inner mitochondrial membrane is impermeable to protons. They cannot simply diffuse back across the lipid bilayer to equalize the gradient. The only way for protons to return to the matrix is through a specific protein channel, and that channel is ATP synthase, the molecular machine that will harness the energy of the proton gradient to synthesize ATP. But before we explore ATP synthase, it's worth pausing to appreciate the elegance of the electron transport chain. The chain operates on the principle of controlled energy release. Electrons from NADH and FADH2 are at a high energy level, and oxygen, the final electron acceptor, is at a very low energy level. If the electrons were transferred directly from NADH to oxygen in a single step, all their energy would be released at once as heat, too abrupt and violent to be useful for ATP synthesis. Instead, the electron transport chain breaks the journey into small steps, each one releasing a manageable amount of energy. This stepwise transfer is more efficient and allows the energy to be captured incrementally as protons are pumped across the membrane at multiple points along the chain. It's an exquisite example of biochemical efficiency, maximizing the useful work that can be extracted from a given amount of chemical energy. And oxygen's role, while it occurs at the very end, is indispensable. Without oxygen to accept the electrons, the entire electron transport chain would back up. The protein complexes would remain reduced, holding onto their electrons with no place to pass them. NADH and FADH2 would accumulate, unable to offload their electrons. The Krebs cycle would slow and stop because it depends on a supply of NAD+ and FAD to accept electrons from its oxidation reactions. Glycolysis would continue for a time, but even it would eventually run into trouble if the cell couldn't regenerate NAD+. The entire process of aerobic cellular respiration is, in a sense, a way of using oxygen to extract energy from glucose. Oxygen is the ultimate electron sink, the final resting place for the electrons that began their journey in the chemical bonds of glucose. Now, with the proton gradient established, with the intermembrane space brimming with protons eager to flow back into the matrix, we come to the heart of ATP synthesis: chemiosmosis and ATP synthase. Chemiosmosis and ATP synthase. The term chemiosmosis was coined by the British biochemist Peter Mitchell in 1961, and it describes the process by which the flow of ions across a membrane down their electrochemical gradient is coupled to the synthesis of ATP. Mitchell's proposal was revolutionary and initially met with skepticism. But over time, experimental evidence confirmed his hypothesis, and he was awarded the Nobel Prize in Chemistry in 1978 for this insight. Chemiosmosis is now recognized as a fundamental mechanism of energy conversion in both cellular respiration and photosynthesis. The key player in chemiosmosis is ATP synthase, a large enzyme complex embedded in the inner mitochondrial membrane. ATP synthase is often described as a molecular motor or a nanoscale turbine, and the analogy is apt. It consists of two main parts: a component called F0, which spans the membrane and forms a channel through which protons can pass, and a component called F1, which extends into the mitochondrial matrix and contains the catalytic sites where ATP is actually synthesized. The two parts are connected by a stalk that rotates as protons flow through F0. The structure of ATP synthase is intricate. The F0 portion consists of a ring of protein subunits embedded in the membrane, forming a rotor. Protons from the intermembrane space flow through channels in F0, entering the membrane and then exiting into the matrix. As they flow, they cause the ring to spin. Much like water flowing through a water wheel causes it to turn. This rotation is then transmitted through the central stalk to the F1 portion, which sits like a mushroom cap atop the stalk. The F1 portion contains 3 pairs of subunits arranged around the stalk. These subunits undergo conformational changes as the stalk rotates, and these changes drive the synthesis of ATP. Here's how it works. Each of the 3 catalytic sites in F1 can exist in one of 3 conformations: open, loose, or tight. When a site is in the open conformation, it can bind ADP and inorganic phosphate. As the stalk rotates, driven by the flow of protons through F0, the site transitions to the loose conformation, where the ADP and phosphate are held in position. Further rotation brings the site into the tight conformation, where the ADP and phosphate are squeezed together so forcefully that they bond, forming ATP. Yet another rotation shifts the site back to the open conformation, where the newly formed ATP is released. Each complete 360-degree rotation of the stalk results in the synthesis of 3 molecules of ATP, 1 at each of the 3 catalytic sites. It's a continuous cycle, elegant and efficient, powered entirely by the flow of protons down their gradient. The beauty of this mechanism lies in its coupling of 2 processes that might seem unrelated: the flow of protons, driven by their concentration and electrical gradients, and the chemical reaction that joins ADP and phosphate to form ATP. ATP synthase acts as a transducer, converting the potential energy stored in the proton gradient into the chemical bond energy of ATP. No covalent bonds in the substrate molecules directly provide the energy, as they do in substrate-level phosphorylation. Instead, the energy comes from the collapse of the proton gradient, and ATP synthase is the machine that captures this energy and puts it to work. The efficiency of this process is remarkable. It's estimated that for every 4 protons that flow through ATP synthase, 1 molecule of ATP is synthesized. Given the number of protons pumped across the membrane by the electron transport chain, and accounting for some protons that leak back across the membrane or are used for other purposes, the complete oxidation of one molecule of NADH results in the synthesis of about 2.5 molecules of ATP, while the oxidation of one molecule of FADH2 produces about 1.5 ATP. These are not whole numbers because ATP synthesis is not a fixed stoichiometry. It's a continuous process driven by the proton gradient, and the exact yield can vary depending on conditions within the cell. Let's now tally up the total ATP yield from the complete oxidation of one molecule of glucose, combining all the stages we've discussed. Glycolysis produces a net gain of 2 ATP directly, plus 2 NADH. If those NADH molecules donate their electrons to the electron transport chain, and assuming they do so via the malate-aspartate shuttle, which is common in many cell types, they contribute about 5 ATP. The transition stage, pyruvate oxidation, produces 2 NADH per glucose, contributing another 5 ATP. The Krebs cycle produces 2 ATP directly, 6 NADH contributing about 15 ATP, and 2 FADH2 contributing about 3 ATP. Adding it all up, we get approximately 30 to 32 molecules of ATP per glucose, depending on the exact efficiency of the process and which shuttle system is used to transport the electrons from cytoplasmic NADH into the mitochondria. Older textbooks often cite a theoretical maximum of 38 ATP per glucose, but this number was based on outdated assumptions about the stoichiometry of proton pumping and ATP synthesis. Modern estimates, based on more precise measurements, converge on a range of 30 to 32 ATP per glucose under optimal conditions. Even this is a theoretical maximum. Real-world efficiency is often somewhat lower because not all protons pumped by the electron transport chain pass through ATP synthase. Some leak back across the membrane through other pathways, and some are used by the mitochondria for other purposes, such as transporting molecules into the matrix. Still, even at 30 or 32 ATP per glucose, cellular respiration is astonishingly efficient compared to the 2 ATP produced by glycolysis alone. Aerobic respiration extracts about 15 to 18 times more energy from glucose than anaerobic processes. This efficiency is what makes complex, multicellular, energy-hungry organisms like animals possible. Without the electron transport chain and chemiosmosis, without oxygen to serve as the final electron acceptor, life would be confined to simpler forms with much lower energy budgets. The process of chemiosmosis illustrates a profound principle in biology. Energy can be stored not just in chemical bonds, but in gradients across membranes. The proton gradient is a form of potential energy, analogous to water held behind a dam or a compressed spring. And just as water flowing downhill can turn a turbine to generate electricity, protons flowing down their gradient through ATP synthase generate ATP, the cell's energy currency. This principle is not unique to mitochondria. It's also used in chloroplasts during photosynthesis, where light energy is used to create a proton gradient that drives ATP synthesis, and in bacteria, where the plasma membrane serves the same function as the inner mitochondrial membrane. The complete energy accounting. Now that we've followed glucose through all the stages of cellular respiration, from its initial breakdown in glycolysis to the final synthesis of ATP by ATP synthase, it's worth taking a step back and looking at the complete energy accounting. Where does the energy in a glucose molecule reside, and how is it extracted and transformed into ATP? Glucose is a stable molecule, and its stability reflects the strength of the chemical bonds holding its atoms together. The carbon-carbon and carbon-hydrogen bonds in glucose store potential energy originally captured from sunlight by photosynthetic organisms. When those bonds are broken during cellular respiration, the energy is released. However, energy is not destroyed but transformed, and cellular respiration is the process of transforming the chemical bond energy of glucose into the more usable form of ATP. The transformation proceeds in stages, each extracting a portion of the available energy. Glycolysis breaks the 6-carbon glucose into 2 3-carbon pyruvate molecules, capturing a small amount of energy in the form of 2 ATP and 2 NADH. This represents roughly 2% of the total energy in glucose. The transition stage and Krebs cycle further dismantle the carbon skeleton, releasing carbon dioxide and capturing more energy in the form of additional NADH, FADH2, and a small amount of ATP. By the time the Krebs cycle is complete, the glucose has been fully oxidized, all its carbon atoms released as carbon dioxide, and the majority of its energy transferred to electron carriers. The electron transport chain and chemiosmosis then convert the energy held by NADH and FADH2 into ATP. Electrons flow through the protein complexes of the electron transport chain, releasing energy in small increments as they descend to lower energy levels. This energy is used to pump protons across the inner mitochondrial membrane, creating a gradient. The collapse of that gradient as protons flow back through ATP synthase drives the synthesis of ATP. This final stage captures the bulk of the glucose molecule's energy, producing about 26 to 28 ATP per glucose, depending on conditions. The overall efficiency of cellular respiration, defined as the proportion of glucose's energy that ends up stored in the chemical bonds of ATP, is approximately 34% under optimal conditions. The remaining 66% or so is released as heat, which is not wasted. In warm-blooded animals like humans, this heat helps maintain body temperature, keeping the enzymes and cellular processes functioning at their optimal rates. Even in organisms that don't regulate their body temperature, the heat produced by cellular respiration is a natural consequence of the second law of thermodynamics, which states that no energy transformation is perfectly efficient. 34% might not sound impressive until you compare it to human-made machines. A typical internal combustion engine is about 25% efficient at converting the chemical energy of gasoline into mechanical work. Cellular respiration is more efficient than most human technologies, and it operates continuously, quietly, and at body temperature, without the need for high pressures or extreme conditions. It's a testament to billions of years of evolutionary refinement. Of course, not every cell extracts exactly 30 to 32 ATP from every glucose molecule. The actual yield can vary depending on several factors: the efficiency of the proton pumps, the tightness of the inner mitochondrial membrane, the availability of oxygen, and the metabolic demands of the cell all influence the final ATP yield. Cells that are working hard and consuming ATP rapidly may have a slightly different efficiency than resting cells. And under certain conditions, such as when proton leak across the membrane is high, the yield may be lower. There are also proteins called uncoupling proteins that can deliberately short-circuit the proton gradient, allowing protons to flow back into the matrix without passing through ATP synthase. This might seem wasteful, but it serves a purpose. In brown adipose tissue, a type of fat found in newborns and hibernating animals, uncoupling proteins dissipate the proton gradient as heat, generating warmth without producing ATP. This is called non-shivering thermogenesis, and it's an important way for small mammals and infants to maintain body temperature when they can't shiver. Even in adults, some heat production from mitochondria likely occurs through uncoupling, contributing to overall metabolic rate. The story of glucose and cellular respiration also highlights the interconnectedness of metabolic pathways. While we've focused on glucose as the starting material, cellular respiration can process other molecules as well. Fats, proteins, and other carbohydrates can all be funneled into the pathways of cellular respiration at various entry points, contributing to ATP production. This metabolic flexibility is essential for survival, allowing organisms to extract energy from whatever nutrients are available. Beyond glucose, alternative fuels. While glucose is the primary fuel for cellular respiration in many cells, especially in the brain and red blood cells, it's far from the only molecule that can be used to generate ATP. The human body is metabolically flexible, capable of extracting energy from fats, proteins, and other carbohydrates, depending on what's available and what the cell needs. The pathways we've explored, glycolysis, the Krebs cycle, and the electron transport chain, are designed to accommodate a variety of fuel molecules, each of which can be converted into intermediates that feed into these central pathways. Fats, or more precisely lipids, are the most energy-dense molecules we consume. A gram of fat contains more than twice the energy of a gram of carbohydrate or protein, making fats an excellent long-term energy storage molecule. When the body needs to tap into its fat reserves, perhaps during prolonged exercise or fasting, fat molecules are broken down in a process called lipolysis. Each fat molecule, typically a triglyceride, is split into glycerol and 3 fatty acid chains. The glycerol can be converted into an intermediate of glycolysis and fed into the pathway at that point. The fatty acids, however, take a different route. Fatty acids are broken down in a process called beta oxidation, which occurs in the mitochondrial matrix. Beta oxidation is a cyclical process that sequentially removes 2 carbon units from the fatty acid chain, each time producing 1 molecule of acetyl-CoA, along with 1 NADH and 1 FADH2. A typical fatty acid, such as palmitic acid with 16 carbon atoms, yields 8 molecules of acetyl-CoA through beta oxidation. Each of these acetyl-CoA molecules can then enter the Krebs cycle, just as the acetyl-CoA from glucose does, and be fully oxidized to carbon dioxide, generating additional NADH, FADH2, and ATP. The energy yield from a fatty acid is substantial. The 8 acetyl-CoA molecules from palmitic acid, passing through the Krebs cycle and contributing to the electron transport chain, along with the NADH and FADH2 produced during beta oxidation, result in the synthesis of over 100 molecules of ATP. This explains why fats are such efficient energy stores. They pack an enormous amount of potential energy into relatively compact molecules. For animals, storing energy as a fat is far more space efficient than storing it as carbohydrates, which is why migratory birds, for example, build up fat reserves before their long flights. Proteins, while primarily structural and functional molecules, can also be metabolized for energy, particularly during starvation or when carbohydrate and fat stores are depleted. Proteins are first broken down into their constituent amino acids, and each amino acid can be processed to yield intermediates that enter the pathways of cellular respiration at various points. Some amino acids are converted into pyruvate, allowing them to enter the pathways just as glucose does. Others are converted into acetyl-CoA, or directly into intermediates of the Krebs cycle, such as alpha-ketoglutarate or oxaloacetate. The specific pathway depends on the structure of the amino acid, but the end result is the same. The carbon skeleton of the amino acid is oxidized, and its energy is captured as ATP. This metabolic flexibility extends to other carbohydrates as well. Starches and glycogen, the storage forms of glucose in plants and animals respectively, are broken down into glucose molecules that feed directly into glycolysis. Disaccharides like sucrose and lactose are split into their component sugars, which can then be metabolized. Even more complex carbohydrates, such as cellulose in the diets of herbivores, can be broken down by specialized enzymes or gut bacteria, ultimately yielding sugars that enter the pathways of cellular respiration. The Krebs cycle, in particular, serves as a metabolic hub, a central junction where molecules from the breakdown of fats, proteins, and carbohydrates converge. Its intermediates can be drawn off for biosynthesis, providing starting materials for the production of amino acids, nucleotides, and other essential molecules, or they can be fully oxidized for energy. This dual role, both catabolic, breaking down molecules for energy, and anabolic, building up molecules for growth and repair, makes the Krebs cycle one of the most versatile and important pathways in all of metabolism. The ability to switch between different fuel sources is vital for survival. After a meal rich in carbohydrates, your body uses glucose as its primary fuel. As glucose levels drop, the body begins to mobilize glycogen stored in the liver and muscles. If fasting continues, or during prolonged exercise, the body shifts increasingly to fat metabolism, burning fatty acids and, in extreme cases, producing ketone bodies from fat breakdown that can be used by the brain as an alternative to glucose. During starvation, proteins are eventually broken down to provide amino acids that can be converted to glucose through gluconeogenesis, a pathway that synthesizes glucose from non-carbohydrate precursors, ensuring that organs like the brain, which rely heavily on glucose, continue to receive fuel. This metabolic flexibility is one of the hallmarks of complex life. It allows organisms to survive in changing environments, to endure periods of scarcity, and to adapt their metabolism to the demands of different activities and physiological states. And it all relies on the same basic machinery, the pathways of cellular respiration, honed and refined over billions of years of evolution. When oxygen runs out, fermentation. While aerobic cellular respiration, with its electron transport chain and chemiosmosis, is highly efficient, it has one critical requirement: oxygen. Oxygen serves as the final electron acceptor in the electron transport chain, and without it, the entire process grinds to a halt. But life doesn't stop when oxygen runs out. Cells have backup pathways that allow them to continue producing ATP, albeit at a much lower rate, even in the absence of oxygen. These pathways are collectively known as fermentation. Fermentation is essentially glycolysis followed by a simple reaction that regenerates NAD+ so that glycolysis can continue. Remember, glycolysis produces two molecules of NADH when it oxidizes glucose to pyruvate. Under aerobic conditions, this NADH delivers its electrons to the electron transport chain, where it's oxidized back to NAD+. But without oxygen, the electron transport chain can't operate, and NADH accumulates. If NAD+ is not regenerated, glycolysis will stop because there will be no NAD+ available to accept electrons in the oxidation steps. Fermentation solves this problem by using pyruvate itself, or a derivative of pyruvate, as an electron acceptor, allowing NAD+ to be regenerated and glycolysis to continue. There are two main types of fermentation: lactic acid fermentation and alcoholic fermentation. Both begin with glycolysis, and both produce NAD+ from NADH, but they differ in what happens to the pyruvate and what end products are formed. Lactic acid fermentation occurs in your muscle cells during intense exercise. When you sprint or lift something heavy, your muscles demand ATP faster than your circulatory system can deliver oxygen. Glycolysis ramps up, producing ATP rapidly without requiring oxygen. But this generates NADH that cannot be oxidized by the electron transport chain because oxygen is scarce. To regenerate NAD+, the pyruvate produced by glycolysis accepts electrons from NADH, converting pyruvate into lactic acid, or more precisely, lactate. This reaction is catalyzed by the enzyme lactate dehydrogenase, and it allows glycolysis to continue operating, producing 2 ATP per glucose even in the absence of oxygen. The lactic acid accumulates in the muscles and can cause a burning sensation, the familiar feeling of muscle fatigue during intense exercise. However, lactic acid itself is not the primary cause of long-term muscle soreness. That's due to microscopic damage to muscle fibers. Once exercise stops and oxygen becomes available again, the lactic acid is transported to the liver, where it can be converted back to pyruvate and either oxidized through aerobic respiration or converted back into glucose through gluconeogenesis. Lactic acid fermentation is not limited to animal muscle cells. Certain bacteria, such as those used in yogurt and cheese production, also carry out lactic acid fermentation. These bacteria ferment the lactose sugar in milk, producing lactic acid, which lowers the pH and causes the milk proteins to coagulate, creating the texture and tangy flavor of yogurt and cheese. Alcoholic fermentation, on the other hand, is carried out by yeast and some types of bacteria. In this process, pyruvate is first decarboxylated, releasing carbon dioxide and forming a 2-carbon molecule called acetaldehyde. The acetaldehyde then accepts electrons from NADH, reducing it to ethanol and regenerating NAD+. This allows glycolysis to continue, producing 2 ATP per glucose. Humans have exploited alcoholic fermentation for thousands of years to produce beer, wine, and bread. In beer and wine production, yeast ferments sugars, producing ethanol and carbon dioxide. The ethanol is the intoxicating component of the beverage, while the carbon dioxide is typically released, although in sparkling wines and beer, some of it remains dissolved, creating carbonation. In bread making, yeast ferments sugars in the dough, and the carbon dioxide produced causes the dough to rise, creating the light, airy texture of leavened bread. The small amount of ethanol produced evaporates during baking. Fermentation is far less efficient than aerobic respiration. Glycolysis produces only 2 ATP per glucose, compared to the 30 or so ATP produced when glucose is fully oxidized through aerobic respiration. The reason is that fermentation leaves most of the energy in glucose locked in the end products, lactic acid or ethanol. These molecules still contain considerable chemical energy in their bonds, energy that can be extracted if they are later oxidized aerobically. In aerobic respiration, glucose is fully oxidized to carbon dioxide and water, extracting nearly all the available energy. In fermentation, the process stops at pyruvate or a derivative, capturing only a small fraction of the energy. Despite its inefficiency, fermentation is essential. It allows cells to survive and function in low oxygen or anaerobic environments. Many microorganisms live in habitats where oxygen is scarce or absent, such as the deep layers of soil, the bottom of stagnant ponds, or the interior of the digestive system. For these organisms, fermentation is the primary or only means of generating ATP. Even in humans, fermentation provides a crucial backup when oxygen delivery can't keep up with ATP demand, allowing muscles to continue working during intense bursts of activity. The existence of fermentation also tells us something about the history of life on Earth. Glycolysis and fermentation are ancient pathways, likely present in the earliest cells, long before oxygen was abundant in the atmosphere. The ability to extract energy from glucose without oxygen was essential for life in the anoxic environment of early Earth. When photosynthetic organisms began producing oxygen as a waste product, oxygen accumulated in the atmosphere, and new metabolic pathways evolved to take advantage of this powerful electron acceptor. The electron transport chain and chemiosmosis allowed organisms to extract far more energy from their fuel molecules, fueling the evolution of larger, more complex, and more energetically demanding forms of life. But the old anaerobic pathways were not discarded. They remain with us, a reminder of life's deep history, still functioning when needed. The broader context. Why this matters. Cellular respiration is often taught as an isolated topic in biochemistry, a series of reactions and pathways to be memorized. But it's far more than that. It's the process that connects every breath you take to every action you perform, every thought you think. It's the reason you need to eat and the reason you breathe. It's the molecular foundation of your existence, operating silently and continuously in trillions of cells throughout your body. Consider breathing. When you inhale, you draw air into your lungs, and oxygen diffuses from the air spaces into the bloodstream. Red blood cells, packed with hemoglobin molecules, bind the oxygen and transport it through the circulatory system to tissues throughout your body. There, the oxygen diffuses out of the blood, crosses cell membranes, and enters the cytoplasm, eventually making its way into the mitochondria. In the electron transport chain, oxygen accepts electrons and combines with protons to form water. This continuous consumption of oxygen by cellular respiration is why you need to breathe. If you stop breathing, oxygen levels in your blood drop, cellular respiration slows, ATP production falters, and cells begin to die. The brain, which consumes about 20% of the body's oxygen, despite being only 2% of its weight, is particularly vulnerable. Within minutes of oxygen deprivation, brain cells begin to suffer irreversible damage. The carbon dioxide produced by cellular respiration follows the reverse path. It diffuses out of the mitochondria, out of the cells, into the bloodstream, and is carried back to the lungs, where it diffuses into the air spaces and is exhaled. The rate of breathing is actually regulated primarily by carbon dioxide levels, not oxygen levels. Sensors in the brain and major blood vessels monitor the concentration of carbon dioxide in the blood, and when it rises, signaling increased cellular respiration and ATP production, the brain signals the respiratory muscles to breathe faster and deeper, expelling the carbon dioxide and bringing in more oxygen. This elegant feedback loop ensures that your breathing rate matches your metabolic needs, speeding up during exercise and slowing during rest. The food you eat provides the fuel for cellular respiration. Carbohydrates, fats, and proteins are broken down during digestion into smaller molecules, glucose, fatty acids, and amino acids, which are absorbed into the bloodstream and delivered to cells. Inside the cells, these molecules enter the pathways of cellular respiration, where their chemical energy is extracted and converted to ATP. The more active you are, the more ATP you need and the more fuel you must consume. Athletes who have high energy demands require diets rich in calories to sustain their training and performance. Conversely, if you consume more fuel than you use, the excess is stored primarily as fat to be drawn upon later when needed. Cellular respiration is also intimately connected to body temperature. The inefficiency of the process, the roughly 66% of glucose's energy that is released as heat rather than stored in ATP, is not wasted. In warm-blooded animals, this heat is essential for maintaining a stable body temperature. Your mitochondria are, in a sense, tiny heaters distributed throughout your body, constantly generating warmth as a byproduct of ATP synthesis. This is why you feel warmer during exercise. Your muscles are consuming ATP rapidly, which means cellular respiration is running at full speed, producing not only more ATP but also more heat. Disruptions to cellular respiration can have serious consequences. Many poisons and toxins exert their effects by interfering with cellular respiration. Cyanide, for example, binds to complex V4 in the electron transport chain, preventing it from transferring electrons to oxygen. This halts the electron transport chain, stops ATP production, and can be lethal within minutes. Carbon monoxide binds to hemoglobin far more tightly than oxygen does, reducing the blood's ability to carry oxygen to tissues and impairing cellular respiration. Even chronic conditions can affect cellular respiration. Mitochondrial diseases, caused by mutations in mitochondrial DNA or nuclear genes encoding mitochondrial proteins, disrupt the function of the electron transport chain or other components of cellular respiration. These diseases can affect any organ, but they often impact high-energy demand tissues like the brain, heart, and muscles, leading to symptoms such as muscle weakness, neurological problems, and heart failure. On the other hand, understanding cellular respiration opens the door to potential therapies. Researchers are exploring ways to enhance mitochondrial function, to protect mitochondria from damage, or to replace damaged mitochondria as treatments for diseases ranging from neurodegenerative disorders to metabolic syndromes. The mitochondria are increasingly recognized not just as powerhouses but as key players in cellular signaling, apoptosis or programmed cell death, and the regulation of metabolism. They are dynamic organelles that respond to the cell's needs, changing their shape, fusing and dividing, and even communicating with the nucleus to adjust gene expression. Cellular respiration also provides a window into evolution. The pathways of glycolysis, the Krebs cycle, and the electron transport chain are remarkably conserved across different species, from bacteria to plants to animals. This suggests that these pathways evolved early in the history of life and have been retained because they are so effective. The similarities between mitochondria and bacteria in structure, DNA, and biochemistry support the endosymbiotic theory, the idea that mitochondria originated as free-living bacteria that were engulfed by ancestral eukaryotic cells. This ancient partnership was one of the most important events in the evolution of complex life, enabling the emergence of organisms with high metabolic rates and large, energy-hungry cells. And there is a poetic quality to cellular respiration, a sense of connection and continuity. The oxygen you breathe was produced by photosynthetic organisms, plants, algae, and cyanobacteria, which use sunlight to split water molecules, releasing oxygen as a byproduct. You take in that oxygen and use it to extract energy from glucose, itself a product of photosynthesis. In the process, you release carbon dioxide and water, which can be used by photosynthetic organisms to make more glucose and oxygen. This cycle links respiration and photosynthesis, connects animals and plants, and ties the living world together in a continuous exchange of matter and energy. The carbon atoms in your body were once part of the atmosphere, then incorporated into a plant, then consumed by you. The water molecules you exhale were formed in your mitochondria from oxygen you breathed and hydrogen stripped from food molecules. Everything is connected, recycled, flowing through the great cycles of life. Closing reflection. And so, as we reach the end of our journey through cellular respiration, let's take a moment to reflect on what we've discovered. We started with a simple molecule, glucose, a 6-carbon sugar carrying energy captured from sunlight. We followed it through glycolysis in the cytoplasm, where it was split into 2 pyruvate molecules, yielding a small amount of ATP and beginning the harvest of electrons. We watched as pyruvate entered the mitochondria and was converted to acetyl-CoA, ready to enter the Krebs cycle. We traveled through the Krebs cycle, witnessing the complete dismantling of the carbon skeleton, the release of carbon dioxide, and the accumulation of electron carriers, NADH and FADH2, loaded with high-energy electrons. Then we descended into the inner mitochondrial membrane, where the electron transport chain operates, passing electrons through a series of protein complexes, pumping protons, and creating a gradient. We saw how oxygen, the oxygen you breathe, serves as the final electron acceptor, allowing the process to continue. And finally, we marveled at ATP synthase, the molecular turbine, spinning as protons flow back across the membrane, harnessing the energy of the gradient to synthesize ATP, the universal energy currency of life. From one molecule of glucose, the process generates 30 or more molecules of ATP, enough to power countless cellular activities. And this is happening continuously in every cell of your body, every moment of every day. Right now, as you listen or read these words, trillions of glucose molecules are being dismantled. Trillions of electrons are flowing through electron transport chains. Trillions of protons are rushing through ATP synthase, generating the energy that keeps you alive, that powers your heartbeat, your breathing, your thoughts. It's a miracle, not in the supernatural sense, but in the sense of being awe-inspiring, intricate, and beautiful. It's a process that has been refined over billions of years of evolution, optimized for efficiency, reliability, and flexibility. It's a process that connects you to all other living things, that links you to the sun through the energy captured by plants, that ties you to the earth through the oxygen produced by photosynthetic organisms. It's a process that makes you, in a very real sense, a participant in the grand cycles of the planet, a node in the vast web of life. You don't need to understand every detail to appreciate the wonder of it. Even the broad strokes, the transformation of glucose into carbon dioxide and water, the coupling of oxidation to phosphorylation, the flow of electrons and protons, reveal something profound about the nature of life. Life is not a static thing but a process, a flow of energy and matter, a constant building up and breaking down, a dynamic equilibrium maintained far from thermodynamic equilibrium by the continuous input of energy. As you drift towards sleep tonight, you can rest easy knowing that your cells are taking care of everything. They're extracting energy from your last meal, regenerating ATP, providing the power you need to breathe, to dream, to wake again tomorrow. Cellular respiration is happening without your conscious effort, a background process so reliable you rarely think about it. But now you know its secrets, its stages, its elegance. You know that every breath you take brings in oxygen that will accept electrons in your mitochondria, and every breath you release carries away carbon dioxide, the remnants of glucose fully oxidized. Tonight, as your breathing slows and your thoughts begin to wander, remember that you are not separate from the universe but a part of it, a living system connected to the sun, to the plants, to the atmosphere, and to the ancient history of life on Earth. You are a site of transformation, a place where energy flows, where molecules are broken and reformed, where the chemistry of existence unfolds in trillions of tiny compartments, quietly and continuously. If this journey through the molecular machinery of life has helped you unwind, if it's sparked your curiosity or given you a sense of wonder, consider liking this video and subscribing to the Sleepy Science Society. Your support helps others find their way here, to these calm explorations of the science that shapes our world. Share your thoughts in the comments. Suggest topics you'd like to explore in future episodes, and know that you're part of a community of curious minds seeking understanding, one peaceful evening at a time. Sleep well, and may your mitochondria work effortlessly through the night, generating the energy that sustains you, breath by quiet breath. Good night.